My name is Dirk de Graaf. I'm professor at Ghent University and I am coordinator of the Be Good project. In 2019, my team published in scientific reports about the genetic predisposition of mite resistance. And we discovered that the suppressed mite reproduction trait can be explained by 8 variants in the genome. In the Be Good project, we want to explore what the contribution is of genetics to the health status index. And therefore, we developed a rapid test to screen colonies for the presence of these protective alleles. In this video, my collaborators will explain how this technique works, and we will show some videos about the manipulation in the lab.
So like just explained, we were able to identify single nucleotide variants which were associated with the phenotype suppressed mite reproduction. In this table, you can see an overview of the 8 markers which we identified. Now it is important to develop an assay that allows genotyping, which will pave the way for marker-assisted selection in breeding programs. One of the techniques which can be used for genotyping is the TaqMan assay. In what follows, I will talk you through the basics. Like any PCR, TaqMan assay reactions require a double-stranded template, as well as 2 standard target-specific primers. While 2 primers are all you need for regular PCR, real-time PCR with TaqMan chemistry requires 1 or more sequence-specific oligos, called probes. And of course, we need a Taq polymerase. Probes are quite different from primers in 2 ways. First, they cannot be extended by Taq polymerase since they lack free hydroxyl groups. Second, 2 other molecules are covalently joined to the probes. On the 5' end, a fluorescent molecule is attached, which is known as a reporter. On the 3' end, another molecule is bound. This is the quencher, which quenches the fluorescent signal from the reporter under certain circumstances. Here is the intact probe, with reporter in blue and the quencher in yellow. When the probe is now zapped with light, we expect that the reporter gets excited and will emit light or fluorescence. However, because the quencher is in close proximity to the reporter, the energy gets transferred from the reporter to the quencher, and this phenomenon is called fluorescent resonance energy transfer, or FRET. So as long as the probe is intact, there is no permanent increase in fluorescent signal from the reporter. However, during the reaction, the probe becomes degraded, which means that the reporter and the quencher become permanently separated. And when light strikes the reporter now, it will emit a fluorescent signal, and this signal can be detected by the instrument. In the graph, you can see a typical result in which the blue line represents the fluorescent signal of the reporter. Well, here is the reaction. I will guide you step by step. We begin by denaturing our template at high temperature. At 60 degrees, the probe and primers will bind to specific sequences in the template. Then, the polymerase find the primers and start the PCR extension phase by building in nucleotides. When reaching the probe, Taq will degrade the probe by its 5' exonuclease activity, and while doing so, the reporter and quencher will be physically separated, and this will lead to the increase of the fluorescence. So in this graph, you can see the blue line which represents the fluorescence signal of the reporter. So far, the basics of the TaqMan assay. Now it is also possible to use different reporters and probes in one single reaction. So if we want to detect 2 different genotypes, we can develop 2 specific probes which are each linked to a specific reporter. So genotype 1 will generate a blue signal, while genotype 2 will generate a red signal. In our case, genotype 1 is the wild type allele and the reporter used is FAM, while for genotype 2 is the variant allele, and there the probe is linked with Texas red reporter. So if we go back to the aim of our experiment, in which we want to genotype our bees, we can use two different probes which are each specific for one genotype. So for, like said before, genotype 1 is our detection of the wild type allele and will generate a blue signal, while genotype 2 will detect the variant allele and will generate a red signal. When we are genotyping honeybees, different possible outcomes are possible. So if we are genotyping single drones, we can have two possible outcomes. Drones are haploid, which means that they only possess one allele. So we can have a blue signal, which means that our drone possesses a wild type allele, or we can have a red signal, which means that our drone possesses the variant allele. In case we are genotyping single workers, three different outcomes can be generated. So workers are diploid. So if we detect only the FAM signal, this means that two wild type alleles were present in the worker, so our worker is homozygote for the wild type. If only the Texas red was present or generated in the tested sample, this means that the tested worker was homozygote for the variant allele. But if we were able to detect both signals, so a FAM signal and a Texas red signal, this means that our tested worker was heterozygote for the tested allele. So there we have a wild type allele and a variant allele for the tested marker. If it is not possible to analyze single individuals, pooled samples can also be used. The typical result is then the percentage of wild type or variant allele present in the tested sample. And this can be estimated based on the use of a calibration curve in which known amounts of wild type or variant allele were mixed.
Now we will show some hand-on videos in which we will demonstrate how to set up a TaqMan assay. First of all, you need to isolate DNA from the bees you want to genotype. In this particular case, we will genotype a pooled sample of worker bees. We will collect a leg from 30 different worker bees and collect them in a sample tube. Therefore, we use sterile forceps. So after collecting the 30 legs of the different bees, we close the sample tube. We will use the Kia AMP DNA microkit to isolate the DNA, but you can use your method of preference to isolate the DNA. We add lysis buffer and proteinase K to the different samples from which we want to isolate the DNA. After adding the lysis buffer and proteinase K, a short forcing step is included. An overnight incubation step at 56 degrees is needed to ensure the complete lysis of the sample. After the overnight incubation, the samples are centrifuged to remove the drops from inside the lid. After adding ethanol and mixing the samples, the entire lysate is applied to the mini columns. After centrifuging, the collection tube containing the flow-through is discarded, and the mini column is washed twice with washing buffer. Finally, the DNA is eluted in 50 microliter of elution buffer. Now the DNA is ready to be used in the TaqMan assay. Before we can start, the probes and primers for the different SNPs need to be synthesized. The reaction contains next to the probes, primers and DNA, also TNTPs, buffer and a Taq polymerase of your choice. First of all, a master mix is prepared without a template for the specific SNP you want to test. After distributing the master mix over the different sample wells of a microtiter plate, the sample is added. After a short centrifugation step, the plate can be mounted into the machine. And the program is started. After 2 hours, the program is finished and the results are available. So all details are described in the article qPCR assays with dual label probes for genotyping honeybee variants associated with Varroa resistance in BMC veterinary research. To end, I would like to thank all our colleagues at University Ghent and all the partners in the Be Good project.